Assalamualaikum everyone. In today's video, we are going to discuss a whole summary of all the practicals of the biochemistry. So this will be the part one, and in this part one, we will discuss all the experiments which are related to the carbohydrate detection. So for the carbohydrate detection, for example, if you are given a solution and this solution contains some carbohydrates in it, and you have given another solution and which has no sugar in it, so how you will differentiate them? So by these tests you can differentiate them. So first of all, the first test for the detection of carbohydrate is the molish test. So molish test is for the all the carbohydrates, ठीक है. We discuss some other test for the detection of carbohydrates. We have 7 total tests for the detection of carbohydrate. So the molish is the one of all of them which can detect all the carbohydrates. So now let's before we are going to start our video, I request you to please subscribe to my channel, like my videos, and also press the bell icon to get the notification of all the videos which I have uploaded. So thank you very much for subscribing to my channel. Now let's start the lecture. So in the start, they have saying that to detect the presence of carbohydrates in the given solution, you are going to perform your molish test. And always your molish test is going to detect all of the sugars, whether it is monosaccharide, whether it is the disaccharide, whether it is aldehyde, whether it is ketones, it doesn't matter. It detect that solution contain carbohydrate or not. Okay, so what will be the principle of this test? So the principle of this test is very simple. So first of all, you have to hydrolyze your sugar. For example, if you see here, so we have D glucose, a sugar here. So we are what we are doing, we are dehydrogenating it, we are removing water from it. As a result, we gain 5 hydroxy methyl furfural. Okay, so this is the furfural link. If you see, if you remove this compound and this compound, you only see the furan ring. Now furfural, you study the furfural in the furan compound when I am explaining heterocyclic compounds to you. So if you just forgive this part, only just focus on this part, so you will see aldehyde group is attached with the furan ring. So whenever aldehyde is going to attach with furan, your furfural is going to form. Now we are going to add another carbon atom. Okay, because furan consists of only 4 carbon, now we add 5 carbon, and then from 5 carbon we have 5 hydroxy is the OH group. So from this we obtain 5 hydroxy methyl furfural. Furfural is this group. Now we attach a fifth carbon with the furan ring, and after fifth carbon we attach hydroxy group with it. Okay, now what we are going to do, we have for example, this is the D glucose. This D glucose is monosaccharide, but in the solution you could be given a polysaccharide, you could be given a disaccharide. Okay, so whenever you are given and such like solution which are complicated sugars, you first have to add concentrated sulfuric acid to it. Okay, what is the purpose of adding concentrated sulfuric acid? Actually, concentrated sulfuric acid hydrolyzes all the disaccharides and polysaccharides to monosaccharide. For example, if you are given a solution of starch, so your concentrated sulfuric acid is converting this starch into the D glucose. Okay, from D glucose, this process is going to take place. Okay, if you, for example, if you are given a sucrose, so what you're going to do, your concentrated sulfuric acid converts sucrose to D-glucose, and then D-glucose undergo the dehydration process. Okay, and now for the next step, after the preparation of 5 hydroxy methyl furfural, what you have to do, you have to add alpha naphthol. Alpha naphthol is very same structure. First you have to draw alpha naphthalene structure. As you know, all of you know very well that naphthalene has two fused benzene ring. Now to make the alpha naphthol, as its name indicate, that it has OH group. So whenever you are going to add a OH on the one benzene ring, it will going to form alpha naphthol. So now you are going to add 5 hydroxy methyl furfural plus 2 molecules of alpha naphthol. Now again dehydrogenation hydration is going to take place and a complex molecule is going to form. So by the condensation with the alpha naphthol, you will gain a purple violet condensation product. So now this complex give a purple color. So due to the appearance of purple color, we indicated that this solution contain carbohydrate or not. So a solution which have no carbon in it give no color, no purple color. Okay, and if a solution has some kind of carbohydrates in it, then it will definitely show purple color to you. So how to remember this structure? This is very simple, just to focus on it. So first you have to take this bond, okay, remove this double bond and oxygen atom. From this oxygen atom you are going to attach this alpha naphthol structure, okay, as it is indicated. And from here part you will remove the hydrogen from this side and you attach another alpha naphthol ring to it. Okay, so two alpha naphthol rings are added to one 5 hydroxy furfural. Okay, so now in the next step, a hydrogen ion releases two electron releases and H3O added and oxygenation occur. After that you will create a double bond here. Okay, in this structure there is no double bond, but here a double bond presents and O and H positive. This is the difference. Okay, so now this is the purple dye. This will produce a purple color and indicated that your carbohydrate is going to present. Now we see how we are going to prepare this molish reagent and what are the apparatus we need, and we see the whole detail, how the solution is going to change its color and how much amount of molish reagent should be added. So these are some related questions. So now let's discuss it. So now we are going to see what are the requirements for this practical and for this test. So you have to need some chemical apparatus and some glasswares and physical apparatus. So for the chemical apparatus, definitely you have to prepare a reagent for the molish reagent. One thing I want to clear in your mind, for all the detection of carbohydrates, protein and lipids, you need a reagent. Okay, and you have to definitely mention the preparation of this reagent, and after that you will show how the procedure is going to occur. Okay, so you have to write the preparation of this reagent. So for this molish reagent you will need 25 gram of alpha naphthalene and 95% of alcohol into it, and then you will dilute it with the 500 ml of alcohol. Okay, so alpha naphthalene, not alpha naphthol. So okay, alpha naphthalene के अंदर when you add alpha 95% alcohol, then it will be converted into alpha naphthol, means the hydroxyl group will going to attach on the naphthalene ring. Okay, so this is the preparation of reagent. Now after preparing this we are going to see the glassware. So the apparatus for this experiment is very simple. You just need two test tubes and test tube stand and pipette is needed. So how the procedure is going to occur? For you have two test tube. On one test tube you will label S for the sample S, and on the other test tube you will label B for the blank. Okay, now you have to add 2 ml of distilled water on the B test tube. For example, this is the B test tube and it has just a distilled water into it. Okay, and this is the S test tube, sample test tube, and you will going to add some sample of carbohydrate which will be given to you, and you add this sample in this test tube. This is the structure of pipette. Okay, now after that you will going to add 2 to 3 drops of molish reagent. Okay, and then you have to thoroughly mix them, and then after that you will have to add it 3 ml of concentrated H2SO4 in it, so that the reaction is going to place, and you have to gently rotate it with your hand. And one thing you have to keep in your mind, don't rotate it very vigorously. Okay, because as you see in the results, only a ring is going to appear whenever you are going to rotate it very fastly. So there is a chances that this ring is going to be distorted, so therefore you should be careful about it. Now we move on to the next page. So now at the last we have some precautions. So you have to take care when you are going to add molish reagent, because if you add some more drops of molish reagent, because you just have to add 2 to 3 drops. If you excessive add molish reagent, then the color of ring changes. For example, we see that the color of ring should be purple, but whenever you add a more amount of molish reagent, the color of ring become green. So therefore you should be careful about it. And then you have the second precaution is that you after the formation of ring, don't shake the test tube, because it can cause distortion of the ring. And then after that you have to add handle H2SO4 carefully, because you know that it is very dangerous chemical. Okay, so the result of this experiment is that you will notice a reddish violet color ring at the junction of two liquids, and this is indicating that your carbohydrate is present. In the last page I have shown how the purple ring is going to be appear. So I think this was enough for you, and if you find this video useful, then go and press the subscribe button. Okay, now we move on to the next lecture. So this is about the second test for the reduction of carbohydrates, and this test is iodine test, and this is for the polysaccharides. Okay, so what is the principle of it? And a polysaccharide only detecting that a polysaccharides is present or not. Okay, this is the iodine test. Okay, so what is the principle of this is that your polysaccharide is helically coiled on each other. Okay, so they absorb the iodine molecules into them. So therefore this test is going to perform only on polysaccharides because of their helically coiled structure. Okay, now your polysaccharide must have 8 plus monosaccharides in their structure, so that they could form the colored complex. And the very important condition for this reaction is that you should need a acidic condition for this experiment, because whenever you are going to use basic effect and basic medium, your secondary valency is going to disrupt it, and your iodine color will not appear. So you should have to use acid, because they protect your secondary valencies, and your iodine absorption will be occur. Okay, and second thing, you have to maintain the temperature, because whenever you heat this solution, it will going to disappear the color of iodine, and whenever you cool on it, it will restore. But accept the dextrine solution, because whenever you heat the dextrine, its properties changes, so therefore it has something distortion and something structural breakdown in it. So in this part, the diagrams of different test tube is given. For example, it contains starch solution. This is glucose, this is glycogen, this is maltose, dextrin, and argiodextrin. So when after we are going to add the iodine reagent, the polysaccharide is only going to show the color. For example, starch is a polysaccharide, so it changes its color. Dextrin changes its color, and glycogen changes its color. But the maltose and glucose and argiodextrin solution not going to change their color, because they are not polysaccharides. So therefore the iodine test is only for polysaccharide. So now we are going to see what kind of material is needed for this experiment. So you should need iodine reagent. You should need 2% carbohydrate solutions, and the carbohydrate solution are glucose, lactose, maltose, glycogen, sucrose, starch, dextrin. These kind of solution you have needed. So now you have to prepare iodine reagent for this experiment. To in order to prepare iodine, you need 2 things. Okay, first you have to need a few crystals of iodine, and second thing, you need a potassium iodide, 2% solution of potassium iodide. You will given potassium iodide, then you will prepare 2% solution of it how you prepared it. You will dissolve 2 gram of potassium iodide in 100 ml, and then you will going to prepare that. And after this, after the preparation of potassium iodide, you will add iodine crystal in it. So how in this way you will prepare your iodine reagent. After this, you will see the procedure. So now what you have to do, you have to take 1 ml of your test solution and add few drops of iodine reagent. So your starch is going to show your blue color, dextrin is going to show red color, and glycogen is going to show purple color. Archodextrin, maltose and glucose is going to show no color because they are not polysaccharides. Now in this test, it has some effect of pH and some effect of temperature. For example, in the effect of pH, it is written that if whenever you are going to add base in it, for example you will add 10% of NaOH, so after adding it, you will see color disappear. So how it is going to happen, because you are going to add base in it, and I have explained to you that base is not suitable for this reagent. And if you are going to add concentrated H2SO4 in it, and then you will see that this color reappears. So therefore this also confirms that your polysaccharide is going to present. Second thing is the effect of temperature. Whenever you are going to heat this solution, you will see that color disappear, but whenever you are going to cool it, the color reappears. But in case of dextrin, the color does not appear because it changes its structural properties. After that, there is some precaution that you know don't have to add any alkaline medium into it. You have to maintain the temperature, because whenever the temperature is changes your solution. So the best temperature for this experiment is 20 degree, and your iodine should not be in excess. Now in that side we have a observation table. For this table we see what kind of solutions we have and what is the quantity of these solution. We take 1 ml of all of these solution. Then after that we have add a few drops of iodine reagent in that, and then at the last we see the color of them. So glucose give no color, archidexin give no color, starch give blue color, maltose give no color, glycogen give purple color, and dextrin give red color. So at the last you will write up the result that iodine test is performed by different carbohydrates, and what compound give what kind of color, you will write it up, and then your experiment is going to be ended. So I hope कि आप लोगों को ये वाला एक्सपेरिमेंट भी समझ आ गया होगा, और अगर आपको फिर भी कोई कंफ्यूजन है तो आप मुझसे कमेंट करके पूछ सकते हैं. अब हम नेक्स्ट एक्सपेरिमेंट की तरफ चलेंगे. ना हमारा नेक्स्ट जो एक्सपेरिमेंट है वो हमारा बारफॉइड टेस्ट के ऊपर है, और ये भी एक मेथड है हमारे कार्बोहाइड्रेट्स को डिटेक्ट करने का, और ये भी बहुत ज़्यादा सिंपल है. ठीक है, तो अब हम इसमें देख लेते हैं. तो एक्चुअली जो हमारा पहला आयोडीन था वो किसके डिफरेंस के लिए था? यस, वो सिर्फ पॉलीसेक्राइड्स के लिए था. और मॉलिश एजेंट किसके लिए था? Yes, you are right. Molish reagent is only for all of the carbohydrates, sugary compound. Okay. Now for the Barfoed reagent, it is only to differentiate monosaccharides from the disaccharide. Okay. So and it is always going to be take place in a acidic medium. Okay. So you have to mention the conditions for every experiment. And after that you should need a Barfoed reagent for this, and you have to add up the principle for this. Now what is the Barfoed reagent? Actually it is a cupric acetate ion which convert or reduce into the cuprous ion. Okay, your cupric ion is going to change into cuprous. So this is the reduction reaction. And now we see what is the principle of it. So the principle of it is that your monosaccharide react very fastly and give the positive results in just 3 minutes, but your disaccharide also give the positive test, but they take a longer time. They will take 2 to 3 and 5 minutes. So basically it is a reduction process, and just the difference between them is that both of these compounds are going positive test, but just the difference of time. So when you are going to perform this experiment, you should need a stopwatch for it. So in the diagram you will see I have taken the two test tube. So in the two test tube you will see the ready precipitate appear at the bottom as well as at the surface in both of these test tubes. So this red ppt appear after 3.5 minutes, so it means it is a monosaccharide. But if it is going to appear after 15 minutes or after 10 minutes, so it is going to indicate that it is a disaccharide. And if there is no appearance of a red ppt, it is going to show that it has no monosaccharide as well as no disaccharide in it. So this was the main summary of the Barfoed test. So why your red ppt is going to be appear at the bottom? This is due to the this ion, this cupric oxide ion. It has the color of brick red ppt. So whenever your sugar is going to react with the cupric ion, it is going to produce cuprous oxide, and this is the reduction reaction. So now the reagent used for this experiment is 2 cupric acetate ion, okay, and then the second thing is glacial acetic acid. So for the preparation of this reagent, you will take 24 gram of this copper acetate and 2.5, usually you can say 2 ml, okay, 24 ml and 22 ml of these solutions, and then you will stir it, cool it, and then you will add distilled water of 500 ml into it. Now for the procedure, you will take 2 ml of Barfoed reagent and add it into the 1 ml of every solution. You will mix them and you will note the time for them. If they are taking long time, then it will be disaccharide. If it going to show the results in just 3 minutes, so it will be a monosaccharide. So you have to also place the test tube in the water bath. You have to boil it for the 5 minutes and remove the test tube, and then you have to cool it under the running tap water, and then you have to notice the appearance of precipitate. At the last, there is the interpretation. In the interpretation, you will say that red ppt appearance show that your monosaccharide is present within the 3 minutes, and if it takes 3 to 5 minutes, it means it is a disaccharide. Precaution is that your copric ion can interfere with the test tube, so you have to handle the test tube carefully. So this was the third experiment for the detection of carbohydrates, which is the Barfoed reagent. So now let's summarize all of these practicals. So first in the practical we see molish test. Okay, so molish test is for all the carbohydrates. We prepared a molish reagent which is prepared by 25 gram of alpha naphthalene and 95% alcohol. So it is going to produce alpha naphthol, and the whole procedure is explained to you. Then the next is the iodine test. For the iodine test is only for the polysaccharide, and it has some conditions. For example, it cannot take place in the presence of base, it cannot take place due to the high temperature, and it need a constant and a balanced temperature for it. Second thing is that for the Barfoed reagent, we see there is a reduction reaction. Okay, your cupric ion is going to reduce into cuprous oxide ion. Okay, so Barfoed reagent is used to distinguish monosaccharide and disaccharide. Both of these compounds give positive. There's just the difference in their time duration. Monosaccharide react very fastly, while disaccharide take longer time. So you have to mention the time and you have to see what is the difference. Okay, so after that you will easily classify that this test tube has monosaccharide and this test tube has disaccharide in it. So now the next experiment for the detection of carbohydrate is of Seliwanoff's experiment. So Seliwanoff's experiment is very simple and it is just similar to your molish test. Okay, and I hope कि आप लोगों को molish test बिल्कुल अच्छी तरह से आ गया होगा, क्योंकि मैंने उसकी हर एक चीज़ आपको समझाई है, और स्ट्रक्चर्स के साथ ड्रॉ करने हैं वो भी मैंने आपको समझाया है. ठीक है, आयोडीन और बारफाइड टेस्ट में स्ट्रक्चर्स नहीं थी, इसलिए मैंने आपको नहीं करवाई वो, लेकिन इसके अंदर दोबारा स्ट्रक्चर्स आई हैं और मैं आपको ये बनाना भी सिखाऊंगी. ठीक है, सो ये बिल्कुल सिमिलर है, थोड़ा सा डिफरेंस होगा, बट जिस तरह का प्रोसीजर, जिस तरह का प्रिंसिपल है, तो वो बिल्कुल सेम है मॉलिश टेस्ट के साथ. सो ये आपको तैयार करना इतना ज़्यादा डिफ़िकल्ट नहीं लगेगा. ठीक है, सो हम इस वीडियो में यहीं तक सेरेमनाइज तक देखेंगे. इसके अलावा और भी टेस्ट हैं कार्बोहाइड्रेट्स की डिटेक्शन के लिए, वो फिर मैं नेक्स्ट वीडियो में अपलोड करूँगी पार्ट 2 में. सो पार्ट 2 भी देखना मत भूलिएगा, और चलते हैं अब इस एक्सपेरिमेंट को स्टार्ट करते हैं. सो सबसे पहले हम इसका प्रिंसिपल देख लेते हैं कि प्रिंसिपल क्या है। जिस तरह मॉलिश टेस्ट सब कार्बोहाइड्रेट्स को डिटेक्ट कर रहा था, तो सेलिवानोफ टेस्ट इस तरह नहीं करता, इसका थोड़ा सा डिफरेंस आ जाता है, क्योंकि ये सिर्फ और सिर्फ आपको डिफ्रेंशिएट करेगा एल्डोसिस में और कीटोसिस में। यानी ये देखेगा कि शुगर के ऊपर एल्डिहाइडिक ग्रुप है या कीटोनिक ग्रुप है। ओके, सो इस तरह से ये डिफरेंस करता है, और इस तरह से ये कलर डिफरेंट देता है। ठीक है, जिस तरह से बारफोड टेस्ट था वो उसका डिफरेंस था सिर्फ टाइम का, इसमें डिफरेंस आता है कलर का। सो जो आपको एल्डिहाइड्रोसिस होंगे, मतलब कोई एक ऐसी शुगर आएगी जिसमें एल्डिहाइडिक ग्रुप होगा, वो आपकी लाइट पिंकिश सा कलर देगी। लेकिन अगर आपके पास कोई कीटोनिक शुगर आती है जिसके अंदर कीटोन ग्रुप होगा, तो वो आपके पास ब्रिक रेड कलर या जो डार्क रेड कलर होता है ना, वो शो करेगा। सो अब हम लोग देखते हैं तो कि क्या होता है इसके अंदर और क्या इसका रिएजेंट है। बिल्कुल सिंपल है कि आप लोगों ने फॉर एग्जांपल यहाँ पर आप देखें कि ये मैंने फ्रुक्टोज लिया है, और फ्रुक्टोज आपको पता है कि ये कीटोनिक शुगर है, बिकॉज इसके अंदर कीटोनिक ग्रुप आ जाता है। सब क्योंकि ये कीटोनिक शुगर है, सो इस वजह से ये एक ब्रिक रेड कलर का जो है वो कम्प्लेक्स बनाएगा। तो सबसे पहले क्या करना है, हमने वही करना है, हमने डिहाइड्रेशन करनी है, हमने वाटर निकालना है। और वाटर निकालेंगे हम इस वाली जगह से, ये यहाँ से एक 2H ये और एक ये ऑक्सीजन निकल जाएगा, तो यहाँ पर सिर्फ CH बच जाएगा। ठीक है, इस तरह से हम वाटर यहाँ से निकालेंगे और यहाँ से भी निकालेंगे, क्योंकि हमने यहाँ पर डबल बॉन्ड लगाना है। देख सकते हो यहाँ डबल बॉन्ड है, तो यहाँ से हमारा सिर्फ एक 2H निकलेंगे, और यहाँ से बाकी OH इस तरह से रिमूव होंगे कि यहाँ पर 2 डबल बॉन्ड आ जाएंगे, ये ग्रुप वैसे रहेगा, यहाँ पर एक एल्डिहाइड ग्रुप आ जाएगा। अब आप देख सकते हो कि बिल्कुल सेम चीज़ हो रही है जिस तरह फर्फुरल बना था। फर्फुरल में याद है ना कि वहाँ पर एल्डिहाइडिक ग्रुप आ गया, तो बिल्कुल उसी तरह ही हो रहा है कि ये भी एक फर्फुरल का ही डेरिवेटिव है। ठीक है, लेकिन ये वो नहीं है, 5 हाइड्रोक्सी मिथाइल फर्फुरल नहीं है, ये फर्फुरल का एक डेरिवेटिव है। क्योंकि फर्फुरल हर वो रिंग होगी जिस तरह फर्फुरल में फ्यूरान के साथ हमेशा एल्डिहाइडिक ग्रुप अटैच होगा। ठीक है, सो अब वहाँ पर हमने क्या ऐड किया था, अल्फ़ा नेप्थॉल ऐड किया था। यहाँ जस्ट डिफरेंट ये है कि आप यहाँ पर रेसिनोल ऐड करोगे। रेसिनोल भी एक टाइप का अल्कोहल आ जाता है। ठीक है, और यहाँ पर आप रेसिनोल का स्ट्रक्चर देख सकते हैं, और रेसिनोल के भी 2 मॉलिक्यूल्स डाले हैं, जिस तरह हमने अल्फ़ा नेप्थॉल के 2 मॉलिक्यूल्स डाले थे। सो ये रेसिनोल का स्ट्रक्चर है, इसमें आपने बेंजीन रिंग सिंपली ड्रॉ करना है, एक ऊपर OH लगाना है और एक मेटा पोजीशन पे OH लगाना है। ठीक है, सो इस तरह ये 2 OH जो है ये अटैच होंगे। अब उसके बाद जब ये रेसिनोल के 2 मॉलिक्यूल्स अटैक करेंगे ये फर्फुरल के डेरिवेटिव के ऊपर, तो इस तरह का कम्प्लेक्स जो है वो स्ट्रक्चर बन जाएगा। ये आप यहाँ पर देख सकते हैं। तो इसमें जस्ट क्या करना है कि आपने ये वाला जो मॉलिक्यूल है इसको थोड़ा सा टिल्ट करना है। इसी मॉलिक्यूल को मैंने उठा के ऐसे रख दिया, देख सकते हो, और यहाँ का एल्डिहाइड ग्रुप जो है वो आगे जाके यहाँ पर अटैक करेगा। अब आपके ये जो 2 रेसिनोल है ना, ये आपस में मिलते हैं। यहाँ पर आप देख सकते हो कि दोनों रेसिनोल के रिंग्स हैं, और इन्होंने आपस में एक दूसरे को एक और बेंजीन रिंग बना के एक दूसरे को अटैच कर लिया है। ठीक है, सो इस तरह से ये एक रिएजेंट और एक कम्प्लेक्स बन जाता है जिसका कलर क्या है, चेरी रेड कलर है। ठीक है, डीप चेरी रेड कलर है। सो अब हम नेक्स्ट पेज पे चलते हैं। सो यहाँ पर ऊपर ही आपको जो है पूरे टेस्ट हमारे प्रैक्टिकल की एक जनरल इक्वेशन दिखा दी गई है कि आपने फ्रुक्टोज डाला। अच्छा, एक और चीज़ आप लोग यहाँ पर मेंशन कर सकते हैं कि वहाँ पर हमने H2SO4 डाला था। ठीक है, ताकि हम शुगर को कम कर सकें, नीचे ला सकें, या उसको फर्फुरल में भेज सकें। तो यहाँ आप HCl डालेंगे और आपका फर्फुरल आ जाएगा। अब आपने अगेन फर्फुरल को लेना है और उसको रेसिनोल में डालना है, जबकि वहाँ हमने अल्फ़ा नेप्थोल को यूज़ किया था। तो उसमें डालने के बाद आपको चेरी रेड कलर का कम्प्लेक्स मिल जाएगा। सो प्रिपेयर कैसे करना है, आपने इस रिएजेंट को, आपने रेसिनोल लेना है 0.5 ग्राम, और उसके अंदर डिस्टिल्ड वाटर ऐड करना है, और कंसनट्रेटेड HCl डालना है 33 ml। सो प्रोसीजर आपका इसमें सिंपल सा है कि आपने जो रिएजेंट है वो आपने ज़्यादा लेना है और कंपाउंड सिर्फ 1 ml लेना है। ठीक है, यहाँ देख सकते हो रिएजेंट का 3 ml है और कंपाउंड की सिर्फ 1 ml है। सो बॉयल करेंगे आप इसको 30 सेकेंड के बाद आपको चेरी रेड या पिंक कलर मिलेगा, ठीक है। और पिंक कलर की सिर्फ रिंग मिलेगी, जैसे आप यहाँ से देख सकते हैं कि 2.5 मिनट्स के बाद यहाँ पर एक फेंट पिंक कलर की सिर्फ रिंग मिली है, जो कि ये इंडिकेट कर रही है कि ये एक एल्डो शुगर है। जबकि कीटो शुगर में आप देख सकते हो कि पूरा का पूरा सोल्यूशन ही चेरी रेड कलर का हो गया है। सो और 30 सेकेंड्स के अंदर ही आपको पता लगेगा, ठीक है। तो ये जल्दी रिएक्शन शो करेगा, ये देर से कर रहा है। 30 सेकेंड्स ज़्यादा है, ठीक है, मतलब 1 मिनट भी पूरा नहीं है। और यहाँ पर आप देख सकते हो कि 2 मिनट है, बल्कि 2 मिनट से भी ज़्यादा का टाइम ले रहा है। सो ये चीज़ का डिफरेंस आ जाता है सेल्विनाइट टेस्ट के अंदर। प्रिकॉशंस अगर आप देख रहे हैं तो आपने HCl का ध्यान रखना है, इसकी कंसनट्रेशन जितनी है उतनी ही डालनी है। जैसे इसकी 33 ml है, इससे ज़्यादा डालोगे तो आपका जो कीटोन है वो उस पर असर पड़ सकता है, वो जो है डिस्ट्रॉट हो सकता है। सो इस वजह से आपने इसकी जो कंसंट्रेशन है वो बिल्कुल फिक्स रखनी है, ठीक है। सो ये था आज का एक्सपेरिमेंट। आई होप कि आप लोगों को ये चीज़ समझ में आ गई होगी। अगर आपको फिर भी इसमें कोई कन्फ्यूजन है तो आप मुझसे पूछ सकते हैं, मैं आपको ज़रूर उसका आंसर दूँगी। और अगर आपको वीडियो अच्छी लगी हो तो प्लीज़ वीडियो को लाइक करें, सब्सक्राइब करें और इसे अपने फ्रेंड्स के साथ ज़रूर शेयर कीजिएगा, ताकि वो भी एक्सपेरिमेंट्स को देख सकें और इनको सीख सकें। सो be connected to Medicare channel, and thank you very much for subscribing me and for following my request. I meet you soon in the next lecture. Till up, Allah Hafiz.